Welcome to the second screencast on the Health and Disease unit of f212. This screencast looks at 3 examples of infectious disease. Okay, and the 3 infectious diseases are malaria, AIDS, HIV and TB. And we're going to look at reasons or ways in which this is transferred or communicated between individuals. Okay, malaria is caused by a protozoan called Plasmodium. AIDS, HIV is caused by a virus called a retrovirus, and we'll talk about what retroviruses are later. And TB is caused by a bacteria. Okay, so all of these are obviously pathogens. They're things that cause disease. Going back to our slide about parasites, we could also say that they're parasites as well, in that they live and require the use of human cells and feed off human cells, in the case certainly of both TB and malaria. Okay, but anyway, they're pathogens, okay, so they've got to be able to be transmitted from host to host. And one of the main features of our study of these is to look at different ways in which pathogens can get from host to host. Okay, they also need mechanisms of reproduction, so we'll look at that in each case as well. And they cause damage to host tissues, and we'll see how that occurs in all cases too. And the examples that we've got vectors. Okay, so we'll see that malaria is transferred by a vector, the female Anopheles mosquito. Physical contact, okay, contamination of direct contact of blood and body fluids in AIDS and HIV, okay, and then droplet infection is the mechanism by which tuberculosis is transferred from individual to individual. Okay, so I'm going to look at each disease in a little bit more detail over the next few slides. So the first of these diseases we're going to look at is malaria. Malaria is caused by a protozoan, a protoctistin, which is a single-celled eukaryotic organism. Okay, and it's called Plasmodium. Okay, and as I say, the key feature that we're looking at here is how it's transmitted from host to host. Okay, the host being, in this case, the human being. Okay, and the answer is it's transmitted by a mosquito, a particular species of mosquito called Anopheles, and particularly it's the female Anopheles which acts as what we call the vector. Okay, the vector is the organism which transfers the parasite, which in this case is the Plasmodium, from host to host. Okay, so that we talk about a vector, and in this case the vector is the female Anopheles. Okay, it's got a complicated life cycle. And we're going to look at the life cycle here, and we're going to start off with, as you can start a cycle anywhere in the cycle, but we're going to start the description of the Anopheles being in the blood of a human being and explain how it gets to the blood of another human being. Okay, so starting off with the infected host, contains gametes which have been produced by the Plasmodium. Okay, so male and female sex cells, okay, have been produced by the Plasmodium, and they are in red blood cells in the human being. Okay, that blood is then sucked up by the female Anopheles. Okay, and then what happens is that those sex cells or gametes, they fuse, fuse or fertilization happens in the stomach of the female Anopheles. They then produce zygotes or Plasmodium, okay, and those migrate, they burrow through the wall of the gut of the female Anopheles and migrate to the salivary glands of the Anopheles, okay, and then the Anopheles bites another human being, okay, which is the host, and injects the Plasmodium into the bloodstream of the new host, and then the Plasmodium, first port of call is it goes to the liver, okay, and it gets into liver cells which are called hepatocytes, and they reproduce in those liver cells asexually and eventually burst out of those liver cells, and they move from the liver to red blood cells, okay, where they feed on hemoglobin in the red blood cells, and some of them produce more gametes, okay, and then those gametes can be then sucked up by another female anopheles. So we get back to where we started from. Okay, if I look at that diagrammatically, okay, as I said, we can describe this from anywhere because it's a cycle, but we'll start with where the notes above started with. So here we have a female anopheles sucking up some gametes. Okay, these are gamete cells here. Those are sucked up by the mosquito, and here's the gut of the mosquito. You can see these gametes are then fuse or fertilize to form the plasmodium. The plasmodium then burrows out of the wall of the gut and transfers itself to the salivary gland, and then it then gets injected by the mosquito into another host. Okay, the plasmodium then move in the bloodstream to liver cells, okay, where they reproduce asexually and eventually burst out of these liver cells, and then they migrate in the bloodstream, and they migrate and invade red blood cells where they feed on the hemoglobin of red blood cells, and then eventually burst out and then reinfect more red blood cells. Okay, but in this process, some of these start to produce gametes. Okay, they will undergo meiosis to produce gametes. Okay, and those blood cells which contain gametes are then sucked up by the mosquito. Okay, so that goes full cycle. Okay, we'll come back and look at that diagram when we start talking about how we can try and prevent this this disease. But what I want to do now is just have a quick look at a couple of animations which illustrate what I've just described. So as I say, it's split between the mosquito phase and the human phase. We're going to look at the mosquito phase first of all. So here is our mosquito, which is sucking up some blood from a human. So blood cells which contain our gametes, or gametes we'll call them, into the stomach of the mosquito. The gametes then further develop inside the gut of the mosquito and eventually fuse. So these are fusion then occurs. So I have a male reproductive cell fusing with female reproductive cell there. So fusion occurs, and then this develops into plasmodium, which then burrow through the wall of the gut Then the plasmodium move from the gut towards the salivary glands. So they penetrate into the salivary glands, into the salivary ducts. So there they are in the salivary duct, ready to infect the next host. Okay, so that's the part of the life cycle that occurs inside the mosquito. So remember, there's a phase of fertilization of gametes in the mosquito's stomach. Okay, then they migrate. The zygotes migrate to the salivary glands. Okay, so that's the mosquito phase. Now let's look at what happens in the human. Okay, so we start off by injecting our plasmodium into the host, the next host, okay, the bloodstream of the next host. So here they come from the salivary glands. Okay, so here's the plasmodium in amongst the bloodstream, and the first place it goes to is the liver, okay, where it invades liver cells. So here it's invaded a liver cell, okay, and then it reproduces asexually. Okay, and then they burst out the liver cells, and then they invade red blood cells, okay, where they feed on the hemoglobin and burst out again and infect more red blood cells. And some of the cells will then undergo meiosis to form gametes. Okay, so you have the male and female gametes. Okay, and then they can be sucked up by another anopheles, female anopheles. Okay, so that's the part of the life cycle that occurs inside the human. So remember, there's two bits to that. There is the asexual reproduction that occurs in the liver. Okay, and then there is the feeding phase that occurs in red blood cells, plus the production of new gametes. Okay, so there's the liver phase and the red blood cell phase in the human beings, and in the mosquitoes there's the stomach phase, and then the movement to the salivary glands. Okay, if I go back to the diagram, we can just talk a little bit about prevention of malaria. In preventing disease, what we do is we try and break the life cycle of the pathogen. Okay, that's the key thing to try and do when combating any infectious disease, is to try and break the life cycle of the pathogen. Okay, so there's various ways that we can do that. First of all, we could try and prevent the female anopheles from biting the host. Okay, so we can do that by using insect repellents. Okay, we can do that by using mosquito nets over beds at night and so on, to try and reduce the chances of being bitten by a mosquito. Okay. Secondly, in large urban areas, what they tend to do is they tend to spray insecticides to try and kill as many of these mosquitoes as possible. Okay, they cover watery areas where they lay their eggs with oil, so their eggs don't, or the larvae can't survive. Okay, there's all sorts of strategies. However, there are problems again with insect resistance to pesticides, as that's a problem that they encounter with that strategy. Or we can take anti-malarial drugs, and there's a number of these anti-malarial drugs around. The first one was called quinine, that attacks the plasmodium in the red blood cell phase of its life cycle, and it makes the heme groups in the hemoglobin poisonous to the plasmodium, and therefore kills the plasmodium. Okay, but again, we've had problems. Quinine is more or less ineffective now, because we have got resistant strains of plasmodium which are resistant to quinine, so that's a problem. So we have to develop new anti-malarial drugs. Okay, so there's various ways that we can have of trying to target various bits of the life cycle of the plasmodium. Ideally, it'd be great if we could have a vaccine, but there's problems with vaccine production. Vaccines, first of all, they because the plasmodium spends quite a long part of its life cycle inside cells, the vaccines don't actually wouldn't be able to detect the presence of these parasites, because they're inside cells. Okay. Also, the antigens on the different types, the antigens on these plasmodium here and these plasmodium here are different. Okay, so it's very difficult to actually design a vaccine to target different parts of the life cycle, or this complex life cycle of malaria. Okay, and also there are different strains of malaria, different types of plasmodium, which means you would have to have vaccines for all the different types of plasmodium that cause malaria. So there's all sorts of obstacles in the way of trying to produce a vaccine for malaria. Okay, so that deals with malaria. Okay, the next slide looks at the viral infection, AIDS or HIV. Okay, HIV stands for human immunovirus. Okay, and so we'll look at that. Okay, so and the key thing that these are requiring you to know is the transmission. Okay, so we'll look at the transmission, a little bit about the life cycle of the human immunovirus. Okay, so it's human immunodeficiency virus, which is longer name, human immunodeficiency virus. Okay, it can remain inactive for years in the body, but once it becomes active, it destroys T helper cells. Okay, which help prevent infection. Okay, and then the patient with active HIV develops what's called acquired immune deficiency syndrome, or AIDS, and that manifests itself by the patient succumbing to other opportunist infections. Okay, particularly difficult with patients that have got double infection of TB and AIDS. Okay, but it can be all sorts of other infections that the AIDS sufferer might encounter, and their immune system is unable to combat other pathogens that might be causing disease. Okay, so in terms of transmission, okay, it's direct transmission, so exchange of body fluids, okay, blood to blood, okay, particularly through sexual intercourse is the most common form of transmission. Okay. But also from unscreened blood transfusions. Okay, when HIV first appeared, okay, it was transferred to a lot of people unwittingly by blood products from the blood transfusion service. Obviously nowadays we screen blood for the virus, so it's not so much a problem now, but it was a problem 20 or 30 years ago. Also, a possibility of unsterilised surgical equipment in developing countries. We could pick up the virus if you had unsterilised surgical equipment which was being used upon us. Sharing needles, a problem was quite often a problem with drug users who share needles for injecting themselves with drugs, and needle stick accidents which happen in hospitals where nurses or doctors might accidentally stick a needle into themselves which they've been using on a patient. Okay. Also, across the placenta during birth is another possible way of contracting this virus, and also mother to baby during breastfeeding. Okay, so those are all the direct the contact. So it eventually, in most cases, requires blood to blood or body fluid to body fluid a contact to get HIV from one host to another. Okay, but let's look a little bit at the life cycle of the virus. Okay, so here's a diagram of the human immunovirus. Okay, it's actually what we call a retrovirus because it contains RNA as its genetic material. Okay, so it's very simple. It's got RNA which is packaged up along with some enzymes, particularly reverse transcriptase enzymes. We'll come across these quite a lot in the A-level biology course, reverse transcriptase enzymes and some RNA, and that's all packaged up in a protein coat which is called a capsid. Okay, and then it's surrounded by a phospholipid bilayer envelope, okay, membrane, which it's actually pinched from the previous cell that it actually invaded. Okay, so that's the structure of this retrovirus, the HIV virus. Okay, in terms of its life cycle, this is what happens. Okay, so here's the HIV virus here. Okay, here it is infecting it attaches to a receptor on the side of the host cell, okay, which is a blood cell, the T blood cell. Okay, it then moves into the blood cell. That's in fact what it does is it fuses with the cell membrane. It injects its capsid in, and the capsid then releases the RNA, which is its genetic material, and then the enzyme that was contained within the capsid, this reverse transcriptase, which is an enzyme from the virus itself, converts the RNA into DNA. Okay, the DNA then inserts itself into the host DNA in the nucleus cell of this white blood cell. Okay, and then the white blood cell then undergoes transcription to produce RNA, which then produces the proteins to produce more capsid molecules, okay, to create a new capsid, and then the capsid along with the RNA then moves out of the cell and covers itself in some phospholipid bilayer from the cell surface membrane as it emerges from the cell. Okay, so that's how it works. Again, I've got a little animation of all of that happening.
HIV, the human immunodeficiency virus, is a retrovirus. An HIV particle attaches to the host cell membrane by way of the membrane protein CD4. The cell removes the viral coat as it takes up the virus by endocytosis. Using reverse transcriptase brought in by the virus, the viral RNA makes a complementary cDNA. The viral RNA degrades. The cDNA is the template for a second DNA strand. The double-stranded DNA enters the host cell's nucleus and integrates into a chromosome as a provirus. Eventually, transcription produces viral RNA. The viral RNA travels to the host cell's cytoplasm, where some is reserved for new viral genomes and some is translated, producing new viral proteins. These proteins are assembled to form new capsids and envelopes. The assembled retroviruses bud from the plasma membrane. The new viruses can parasitize more host cells.
Okay, so going back to this diagram here, okay, so this just shows you how the virus reproduces. Okay, so the RNA produces DNA, and then the DNA produces lots and lots of copies of RNA plus the proteins required to make the capsid, and then each bit of RNA, which is surrounded by a capsid, then emerges from the cell. Okay, taking some phospholipid envelope with it. Okay, and then it can infect other cells. Okay, so that's how it reproduces. Okay, going back to looking at how we can control this HIV virus, there are all sorts of drugs. All sorts of drugs can be used, and actually, given our understanding of enzymes inhibitors, it's interesting. The first drug that was ever produced, which was an anti-HIV drug, which was called AZT. Okay, back in the 80s, and it was actually a competitive inhibitor. Okay, so it actually the nucleotides which were required to make the DNA from the RNA, what we did was that we produced an inhibitor for that. Okay, something that looked like a nucleotide but wasn't a nucleotide, which would bind to the reverse transcriptase and prevent further DNA from being produced. Okay, so that was another example, if you like, of a competitive inhibitor used as a drug. Okay, other drugs have been used to try and target this life cycle. So there's actually non-competitive inhibitor drugs which actually also target the reverse transcriptase. Okay, so it's a useful enzyme to target because reverse transcriptase is a viral enzyme. Okay, so this enzyme doesn't do anything naturally within human cells. Okay, so it's only the virus that has this reverse transcriptase. So it's a useful enzyme to be able to target because it can differentiate between viral processes and human processes. Okay, and also the enzymes which were involved in making the capsid, which can also be targeted. These which are protease enzymes. Specific protease enzymes involved in capsid production can also be targeted with drugs. So again, a bit like malaria, what we try and do is we try and target the life cycle. Okay, we try and interrupt this life cycle so we can stop producing more and more HIV viruses to infect more cells. Okay, there are also inhibitors that we can use to try to prevent the HIV from actually gaining entry into these white blood cells in the first place. So So hopefully that gives you an idea about HIV and AIDS. The key thing you need to take away from this is the mode of transmission by exchange of body fluids, blood to blood, or sexual intercourse, blood transfusions, unsterilised surgical equipment, sharing needles. Okay, so moving on to the next slide. So the third disease we're going to look at is TB, or tuberculosis. Okay, and again, the transmission of these diseases are the things that really is of interest here. And in this case, it's called droplet infection, okay, which is the bacteria pass from host to host through droplet infection when person's coughing, sneezing, etc. Okay, and the spread is more likely when you've got overcrowding, okay, poor ventilation in an area. People with poor health are more susceptible to it. People with poor diet are more susceptible to it. Homelessness people are more susceptible to it. Okay, or close proximity to people that have migrated from a TB region. Okay, and also with problems about TB being found in cattle, as you'll know, the whole business about culling badgers, which is so topical at the moment, is to try and prevent the spread of the disease in cattle. I think that it's spread through cattle and cows by badgers. Okay, now it's caused by a bacterium which is called a mycobacterium, which there are a number of species of. Okay, Mycobacterium tuberculosis and Mycobacterium bovis are two species of bacteria that cause tuberculosis. Okay, just as we said, just look a little bit at its life cycle and how it works. First of all, it's a dramatic diagram I've got here. Somebody sneezing, okay, and producing a sort of aerosol of droplets. Okay, and this aerosol of droplets can contain the Mycobacterium tuberculosis or Mycobacterium bovis, whatever it is, and those this aerosol of droplets can be breathed in by another human being and can then infect the lungs. Okay, in terms of how it goes about infecting the lungs, okay, what happens is, going down to this diagram here, it comes down the bronchioles and gets itself into the alveoli, where it actually invades white blood cells that live on the surface of alveoli. Okay, and the white blood cells are unable to, the macrophage cells are unable to digest and break down these tuberculosis bacteria, because the mycobacterium are covered in a protective capsule which prevents the macrophage or the white blood cells from digesting down the mycobacterium. Okay, and what happens is that causes a swelling in the alveoli, and that converts and that grows into something we can actually see, which we call a tubercle. Okay, so the lungs have a characteristic sort of swellings all over them, which are called tubercles. Okay, and then what happens is that these can then reproduce in here. Okay, and then they can get out into the exhaled air and be in droplets, coughed and sneezed out for somebody else to pick up the disease. Okay, so that's a little bit about the life cycle of the Mycobacterium tuberculosis. Okay, a little animation, very short animation here, shows some aspects of it.
Tuberculosis. Tuberculosis is an infectious disease caused by the bacterium Mycobacterium tuberculosis, or tubercle bacillus, that mainly affects the lungs and can spread to other parts of the body. It is characterized by cough, fever, shortness of breath, weight loss, and appearance of tubercles or tiny lumps and caseous necrosis in the tissues of lungs. Tuberculosis.
Okay, so this actually quite nicely, these are these tubercles which I mentioned before. Okay, so these inflammation of these alveoli. Remember the the macrophages that live on the alveolar surface, which you might have had mentioned to you in the f211 course when you did alveolar structure. Okay, they engulf the mycobacterium but can't break it down, and that causes other white blood cells to move in and the inflammation process to occur. So we get inflamed alveoli, which we can actually see, and they become known as tubercles. Okay, and so that's how that occurs. In terms of going back to my diagram here, as I've mentioned previously, targeting this life cycle, trying to break it, because of the capsule that surrounded these mycobacterium tuberculosis, the normal antibiotics, the penicillin type antibiotics, were ineffective against this particular type of bacteria, and it took a while for man to discover an antibiotic which was effective against this. It's an antibiotic that targets the protein synthesizing mechanism inside bacteria, which is slightly different from the protein synthesizing mechanism in normal eukaryotic cells. Okay, ribosomes are slightly different and can be targeted with a particular antibiotic. And the first one that was produced was an antibiotic called streptomycin, which had a huge impact because tuberculosis, certainly in the Western world, had a huge even back in the 1950s, there were a lot of people in this country living in sanatoria around the countryside where they were being treated unsuccessfully for tuberculosis, and then suddenly along came streptomycin, which then was able to eradicate the disease. Okay. Also nowadays what we do is we vaccinate all children against tuberculosis in this country, the BCG vaccination, which hopefully you've all been given, which guards us against the tuberculosis bacteria. So that's the last of the three diseases we've got. This is caused by Mycobacterium tuberculosis, or Mycobacterium bovis. Okay, causes swellings in the lung called tubercles, and can be passed from individual to individual by droplet infection, coughing and sneezing, and the problems can be exacerbated by overcrowding, poor ventilation, poor health, poor diet, homelessness, and close proximity to people that have migrated from a TB region. Okay, so there are, for example, people in London which haven't necessarily had the BCG vaccination, and they live in areas where there's been migration of people from areas, sub-Saharan Africa, for example, where there's lots of TB, and suddenly there's a tuberculosis problem in that region of London. Okay, so that's an ongoing problem for the health service. So moving on to the next slide. So this last slide about three diseases looks at the global impact of the three of them: HIV, TB and malaria. Okay, so dealing with some headlines first of all. 1.2 million people a year infected, killed by malaria, 300 to 500 million people living with malaria. Okay, it's obviously limited only to areas of the world where anopheles can survive. Okay, so that tends to be around the equator. 90% of malaria is found in sub-Saharan Africa. Okay, other issues or concerns we'll deal with later on when we deal with global warming, is that the global warming could actually spread the area of the planet on which the Anopheles mosquito can survive, and if that's the case, the malarial disease, the Plasmodium, could spread to other parts of the world. So even concerns that global warming at the rate it's going at the moment could actually see the Anopheles surviving in southern Europe. Okay, so you could get malaria in holiday in Spain or Italy or places. Okay, so that's a potential thing for the future to be concerned about. Okay, the other next disease to look about is 33.2 million people living with HIV. Kills 2.5 million each year, and again, half of these occur in sub-Saharan Africa. Okay, but it's also spreading rapidly through other areas such as China and Southeast Asia. Okay, tuberculosis, 1% of the population is infected with tuberculosis each year, and 14 million infected worldwide. okay, and then 1.5 million per year die of tuberculosis, okay. So those are all worrying statistics, okay. So so discussing why these areas in sub-Saharan Africa is mentioned for lots of these problems, is the issues where developing countries are more likely to have problems with these diseases. There's lack of health care. So, for example, a lack of a vaccination program for tuberculosis would be a very good reason to have high tuberculosis levels in these areas. Lack of health education. Okay, so the spread of HIV and AIDS could be put down to the fact that people don't understand how to protect themselves from getting those diseases by practicing safe sex practices such as using condoms, etc., or using mosquito nets to protect themselves at night, or using insect repellents to prevent themselves from being bitten by insects, the female anopheles, presenting themselves from getting malaria. Or there could be lack of equipment to prevent the spread. Okay, so there could be lack of condoms in particular areas, or sterile needles in hospitals, or sterile surgical equipment in hospitals, or it could be lack of mosquito nets to prevent yourself from being bitten by the female anopheles. Okay, so that could be lack of equipment. Okay, overcrowding, particularly opposite to tuberculosis. So poor housing, shanty towns, etc. Overcrowding can lead to rapid spread of tuberculosis in people in developing countries. Okay, and then also to consider is the impact that all this has. Obviously, in developing countries, you've got a high death rate, and that high death rate impacts on economic growth. Okay, if people tend to die in their 30s and 40s or whatever, in the middle of their working life, that affects on the economic growth of the country, and therefore there's less funding, okay, to develop your health care and your health education, improve your housing. Okay, so you get into this downward spiral where the situation can't improve. Okay, so those are all aspects about discussing the global impact of these. The World Health Authority tries to monitor the spread of these diseases and tries obviously to provide limited resources it's got to improving health education, improving healthcare in areas which are particularly badly affected with these diseases. Okay, so that concludes our look at the impact of malaria, AIDS, or HIV and TB on the world.